From MicrobeTV, this is Office Hours for Wednesday, November 19th, 2025. I'm Vincent Racaniello, and welcome to your viral corner of the internet, where you can ask any question about viruses and whatever else you want. There may be somebody here that can answer it. Don't know a lot of other things, but with viruses, we can do our best. Hi, everybody. I'm happy to see you. Sorry we missed last week. I was in Toronto where the internet was not very good. We have moderators tonight. We have Les Fabi, Vanity Nutrition, Tom Steinberg. Thank you for joining us. Hope everyone's feeling well. And tell us where you're from tonight. We will shout you out. So what's up for tonight? Well, I'm going to show you some pictures from my travels to Toronto. And then I have a mini lecture on gene editing. Just kind of an overview of the different kinds that are available. It was brought up by a Nature article that we talked about 2 weeks ago, where they said, there's CRISPR-Cas9 and then there's two other methods that might be better. So I thought I would go over those methods with you. It helped me to learn about them too, right? Okay, let's see who's here tonight. Rita is here. We're in Idaho, if I remember correctly. 47F8C. I don't know what it is here. I could actually find out. It's 5 degrees Celsius here in New Jersey. Rita's in the sagebrush-covered northern Great Basin. Les tells us that a new biography called Crick came out this week. On Friday on TWIV, we'll be doing the 1953 paper by Watson and Watson and Crick on the structure of DNA. That should be fun because there's no data in it, as far as I can tell, but it'll still be fun. Hello, viruses, Luna. Rima, nice to see you from Iowa. Tom is in the Oregon Coast Range. 0C. Will is in Changsha, China. 9 degrees C, where it's morning. Hello, John from Minneapolis. But times aren't as tough or cold as what I just saw in episode 3 of the new PBS American Revolution series by Ken Burns. Smallpox makes an appearance. So let's see. That was Les and Tom, and here, Vanity Nutrition. Hope you're feeling better from last time. And I see you and I think of planes. So my plane travel, 4 flights in this past week were all good. No delays. Very nice to see that. EHS Talent is in Lehigh Valley, PA, where it's 2 degrees Celsius. And our favorite nephrologist, Carol, is in Bakersfield, where it's 14C. MN9159 is in DC, and it's chilly. Vanity says, interesting news last week about lupus and EBV. Yes, we're doing the paper Friday on TWIV. Oh, yes. If you took my virology course, you know that EBV is highly prevalent. There's a slide on that. But it's okay, Vanity. I know it's been a while, and it went away. It's no problem. If you don't use it, you lose it. Hello, Questella. Nice to see you. Did you finish reading the parasitic diseases book yet? Lagodica's in Chicago, where it's cold. It's so cold the ice ran away. Whatever it takes to get them to run away is good, because they don't deserve. They don't not deserve. They shouldn't be anywhere. There's no reason for them. Hello, Sandra. Nice to see you. Hazelbra. Oh, I know who it is. Yes. But the name is weird. That's the thing, right? It's changed. From KSA 14C. Nice to see you again. Mike is in Seattle, where it's 48 and cloudy. Elizabeth is in West Virginia. GSD Bio Jonathan is in Encinitas, 14 degrees C. John is in northern Thailand, where Listerine saved my marriage. Now, that's an interesting statement to make. If you would like, we'd love to hear. We'd love to hear about it. Catherine, is in Canada. That's the one flag besides the U.S. flag that I do know. Melinda's in North Carolina, 57 and falling. Christina is in Hershey, PA, a 38F. Pete's in London, where winter has arrived. It will hold around freezing point all night. Some rural areas had a morning flurry of snow. Abdurrahman is in Manhattan. Hello, Abdur. Nice to see you again. Carol really enjoyed TWIV 1271 from Virginia Tech. Hokies Go Wild Part 2. Didn't know Hep B was that prevalent. Very interesting. I went back and listened to TWIV 285. Is that the other Virginia Tech one? Yep, Hokies Go Viral Part 1. So I did an experiment in Virginia Tech because I am really tired of carrying a ton of recording equipment on the road. So I brought a very light tripod, my iPhone, and 2 microphones that clip on, and they wirelessly transmit to a receiver that's plugged into the iPhone. Really good quality sound, and the video on an iPhone is great. It's 4K. And it's hardly any weight. Otherwise, I'm carrying 20, 30 pounds of stuff like I brought to Toronto last week. And it's just a pain in the neck, especially internationally, where I have to walk a bit. So I'm going to Germany in December for a Giant Viruses meeting, and I just picked up a 4-person wireless set that plugs into the iPhone, and I'm going to do that. I'm going to just bring my iPhone and not have to schlep with heavy microphones, heavy wires, heavy camera, heavy lenses, and a recorder, a sound recorder on top of it. So that Virginia Tech, I think, turned out very well. Tom says it's supposed to be a 10C. Okay, got it. On Long Island, it's 5 degrees Celsius. Hey, look at those airplanes. So vanity, when you become a doctor, then, well, they'll probably always think of planes when I see your name, because it's been so long, it gets embedded in your brain. Lori just got back from Austin, Texas in time for office hours. And how about that? A judge overruled the gerrymandering, the redistricting of Texas. Isn't that cool? I really like that. People are starting to get some chutzpah and fighting this corrupt and lying administration. GSD Bio says there was almost a movie or a show made about CRISPR starring J-Lo, face turquoise covering eyes. J-Lo, oh my gosh. She's going to play Jennifer Doudna. I bet Jennifer would like that. So tell me, folks, if they made a movie about me, which will never happen, it's fine. I have no problem with that because I got my own media thing going here. But if they ever made a movie about me, who would they get to play me? Just pop it in the chat. I'm curious what you think. Pete says the theater at the Crick Institute is hosting the recording of a podcast science Q&A series chaired by Professor Brian Cox. We went to one on ancient DNA, humans, and others. That'd be cool. Yeah. Hey, well, what a novel idea, doing podcasts about science. It's incredible, the technology that we have these days, right? Tongue in cheek, yeah. Mark, 11C in Las Vegas. Okay, we'll take a question. Melinda says, somebody told me that it's harder to make vaccines for RNA viruses like Hep C and HIV. Okay, so is this somebody, a vaccinologist or a virologist? If not, then there's no credibility. Do you agree? No, I don't agree at all. It really depends on, well, first of all, just making a vaccine is the same for both RNA and DNA viruses, especially if it's a subunit vaccine, right? Where you got an antigenic protein or a subunit. Make an mRNA vaccine, you can make a virus-like particle vaccine. There's no difference. Now, the only difference occurs if there's antigenic variation, right? It doesn't impact you making the actual vaccine. It impacts what you do afterwards. You have to update the vaccine. So it's good if it's easy to update. But I don't see any reason why it's any different for DNA and RNA viruses. Laura is in Washington, PA, 3C and cloudy. Welcome. Jeff is in Elgin. Is it Elgin or Elgin? Don't know. Illinois, 5C and overcast. Probably Elgin, right? Doug is in Redondo Beach, 15C. Mark Lamont is in the east end of Long Island, 0C. 140,000 subscribers. Thank you. Yes, we hit it sometime in the past week. We just now have a little over that, about 140,100. On our way to 1 million. That's my goal. I want 1 million people listening to great science, okay? Carl is in Gills Rock, Wisconsin, where it's 0 degrees Celsius. Veronika Privyet from Moscow. We've already seen our first snow, -2. Yikes. Well, I go to Toronto, and it's 0 and snowing. Taking the car from the airport, and it's bloody snowing. Let's see, when is it at night that I got in? Yeah, I got in at night. And then the next day it was snowing. And then the next day it was snowing as well. Cloudy and overcast, no sun. A guy from, gave a keynote talk from, what country? Was he from Ghana? He said the weather in Toronto is miserable. And he showed a picture of Ghana with blue skies and sun. Les tells us that George Washington inoculated his slaves, troops, for smallpox. That's correct. It's the inoculation that had a high fatality rate because they were just using regular old smallpox virus. Hat is in the San Fernando Valley, 58F. Scott is in Seattle, 45-ish, about to go paddling in a dragon boat. So what's a dragon boat? Melinda's doing 2 weeks of Thanksgiving. This week with my husband's family and next week with mine. Time to start baking very soon. Pumpkin pie and butternut squash custard. Well, this week I'm releasing the latest episode of This Week in Neuroscience. Is it edited yet? Anyway, it's the neuroscience of overeating. You should listen to our discussion. It's really fun. Make sure I go to a Christmas market. Yes, so I've been to one in Zurich, and they took the train station and made it into a Christmas market. It was amazing, just the food and all the cool stuff, handmade stuff, lovely. And I think I also saw one in Munich once. I'll be passing through the train station in Munich. Definitely will see a Christmas market. Yeah, this is cool. Hello, Steve from Kauai. Going to try and see if my Kaiser otolaryngologist will let me get an HPV vaccine at 75. This is a good question. It may be tough. I think it's a really good idea. Yeah. If I can't get one, I'll just ask Daniel. Rona on Long Island. Hello and welcome. John is in Thailand. Nice, and enjoying the program. That's great, John. Nice to see you. Far away. Sharon is in Lewiston, Maine, where it's 28F. Joshua is in Central Texas, where it's 75S. I'm sorry, F. Okay, so some guesses about who would play me. Ed Norton? Give me a break. I'm not an Ed Norton. That guy's from the Honeymooners, right? Is that who you're talking about? That goofy guy? Yeah. Willem Dafoe, maybe. A younger Billy Crystal. Robert De Niro. That would be funny. That would be funny. Liam Neeson. Yeah, that's interesting. Logotic Seconds, Willem Dafoe. Melinda says Gilbert Godfrey. Keanu Reeves could play you, face blue smiling. That would be interesting. That would be very interesting. Peter Lorre. Oh, yes, Peter Lorre could play me. Gilbert Godfrey was an infectious disease physician, FYI. Neat. William Dafoe, if we're being serious. Are Muppets allowed? Tom Hanks. Mark Rylance. Harvey Keitel. These are all amazing suggestions. Harrison Ford. That would be awesome, right? I guess it's about my age. Yeah, but age doesn't matter, right? You can always make people up. Elgin. Okay, Elgin is in the liquor. Thank you so much, Jeff. I appreciate it. Second for Mark Rylance. Okay, those are very interesting. Okay, so Danny wants to know, how tall are you? I'm 5 feet 7 inches and 150 pounds. Okay, let me go back. I skipped a couple of things here. Yeah, those are all great suggestions. Thank you very much. I'm honored. I don't know who I would want. It doesn't matter what I would want. I mean, yeah, Robert De Niro would be cool, right? The Italian angle. But you know, in Contagion, Elliot Gould played the scientist, right? But I don't think he'd be a match for me, right? Probably not. So Keanu Reeves, you know, I thought the only thing he did well in it was the Matrix series, right? Everything else I saw him in was so wooden. Although lately he's done something which I think is redeeming him. I don't know. Anyway, Pete says the Q&A Crick podcasts are published in video on the Crick Institute YouTube channel. That's cool. I'd love to watch those. I love science history. It's very interesting. All right, thanks, folks, for making your suggestions about who could play me. That's very interesting. Okay, so Razor Jin says, for someone who never took genetics, is all RNA either plus or minus or plus minus? So it can be plus single-stranded plus polarity, single-stranded minus polarity, or double-stranded, which it would have both plus and minus strands, and that's it. There's no more. Tona says, speaking of crick, does anyone go to the Cold Spring Harbor monthly guest talks at the pub? I don't. It's too far. It's like 2 hours, 3 hours for me. Horrible traffic getting, from Manhattan. And so, yeah, I could take a train to Huntington and then take a taxi, right? Yeah. It would take a long time, too. But those would be fun to go to, right? Do you go, Rona? Tona? Sorry. All right, if you want to make degrees, you can do Option Shift 8, right, on a Mac anyway. So Carol asks, on TWIV 1268, Daniel reported 2 vaccinated doctors contracted measles from an unvaccinated child. Will those doctors have immune amnesia? This is a great question because they had mild case, right? So the study that resulted in the conclusion of immune amnesia after measles infection was done in kids with full-blown measles. So we don't know if in a mild case you still get amnesia or not. I would guess there's going to be some immune amnesia, right? But they could do some titers, antibody titers, just to get an idea. That would be an interesting study, actually, for mild measles cases. Good question. Yeah. Okay, the RNA, I presume you're talking about what causes it to be charged. Well, the phosphates, right? They have a negative charge. And those are on the outside of the molecule. So getting RNA into cells is hard because the membrane of the cell is negative stranded. So you have to do something to neutralize the charge. And you usually use chemicals that coat the RNA and make it plus charged so that it binds tightly to the cell. Veronica says, November and December are the worst here, Moscow. Here, barely a single sunny day for weeks, absolutely miserable. Yeah, miserable is what they call Toronto also. David Tennant playing me. Ooh, that's interesting. Let's see. Oh, yeah. Yeah. Scottish. So I don't know. I saw a series with him that took place on the coast of Scotland somewhere. Broadchurch, that's it. And the lady in Broadchurch, right? Olivia Colman. It was a great combo, the two of them. Fabulous. Yeah, so Tennant looks a little bit like me. He's a little skinnier than me, right? That would be cool. And New York Cohen agrees. He likes Dennett. Melinda says, best way not to overeat is intermittent fasting. Well, Melinda's, some people can't. They have genetic defects that make them overeat, and we talk a bit about that on This Week in Neuroscience. And so they can't cut back. And if they fast and they eat, again, they're going to gain weight. If they exercise, it doesn't help them at all. Nevitz says, for your information, Mount Washington in New Hampshire, it's minus 5°C. So Tom writes, you could expand on the question to teach a bit more about HIV and Hep C, how they pose their own challenges in developing an effective vaccine. You certainly have challenges, right? But so do herpes viruses, DNA viruses, and Epstein-Barr virus, right? We don't have a vaccine for any of these. And so I don't think they're specific to the nucleic acid, but it's an interesting mini lecture, yeah. NY Cohen is in White Plains, where it's 37F. NY Cohen, you like the new mayor-elect of New York City? I do. I think he's interesting. Yeah. Ed Norton was he played Art Carney, right? I remember him. I'm thinking of him in black and white, right? But he's not around anymore, right? Costello doesn't know who any of the actors are, but she likes 3 of them. Okay. Oh, sorry. So when Razor was asking about the charge, I thought he was asking about what gives RNA a charge, right? The phosphates. But yes, the plus minus strand is not about charge, as Laurie says. It's just by convention, the plus is that which is translated to protein. In other words, is mRNA. And the minus is the complement, right? It doesn't have anything to do with electrical charge. But RNA is positively charged because, negatively charged because of the phosphates. Okay. One puddles one. Thank you for your support of science communication. I'm sure One Puddles One has a nice name, right, which has been changed. But we appreciate your support of us attempting to tell people about science. Ed Norton was in The People versus Larry Flint, The Italian Job, and many others. Oh, Edward Norton was who I meant, not The Honeymooners. Okay. So there's an Ed Norton in The Honeymooners, and then Edward Norton is a more modern actor. Okay. Got it. The one who commented on the difficulty of making vaccines was an infectious disease physician. Well, that doesn't actually give them much credibility unless they're a clinician, scientist like Daniel Griffin, who, studied immunology. Because, as you know, and to this day, many physicians don't know what they're talking about when it comes to vaccines and antivirals. They just learn what they hear, unfortunately. So, there's no difference between a DNA and an RNA virus vaccine. So you could ask him next time to give you some examples of DNA and RNA virus vaccines where the RNA virus vaccine is harder. Let's see what he has to say. But I could throw back EBV and herpes viruses as equally hard as any RNA virus. Okay. A younger Dustin Hoffman. Hoffman would be okay too, yeah. Yeah. And Hat says if we include 30s actors, Paul Muni, who also played Pasteur. How about Vincent Price? Nah, he's not. I'm not a Vincent Price, no. But I do like Vincent Price. Patricia's in Massachusetts. Hello, Patricia. Anyone have information, Marco Mont, from their country about H3N2 subtype K flu? Subclade. It's a variant, right? And they're always saying it's more virulent. It's just they don't learn anything, this press or anyone. It's not to do with the virus. It has something to do with immunity. Anyway, yeah, a new strain this year could make it worse. Or it could not. The real thing is how it matches with the vaccine, right? And so far, what we know, this is according to Gavi, which got their funding revoked by RFK Jr. stupidly. Gavi is saying it has drifted significantly from this year's vaccine. So, I mean, that in itself would cause disease, right? Because you're not getting as much protection from the vaccine against disease. Tona says, Ethan Hawke. I didn't see Blue Moon. Ethan can be anyone. Keanu Reeves is currently on Broadway in a new play. Yeah, it's getting good reviews. That's why I said I think he's got some revival there, because the stuff he did after Matrix all sucked, or at least they got poor reviews. Vincent, Ed Norton is an actor, not the character you are thinking of from The Honeymooners. So that's the thing. I'm sorry. Ed Norton was the character played by Art Carney. That's right. Some stupid me. Sorry. I don't think he would be good at playing me because his character was goofy, right? It's funny. You see, don't ask me about movies or TV shows. I don't know anything about these people. Who's going to play Amy? Okay, so that's a good question. Now, tell us who would play Amy, who many of you know. She was on, I think Amy Winehouse should play Amy, but she wasn't an actor, right? But she's still an actor as a musician, right? Rita read that a person in Western Washington had H5N5. What's that? It's a influenza virus with an H5 hemagglutinin, right? And an H5 neuraminidase. It happens. I've read papers about them. Yeah, first case ever, human case of H5N5. Yeah. So, where did they come from? So the person who got it has a backyard flock. So ostensibly, it's a avian influenza virus, right? Helen, hello from Research Triangle Park. Nice to see you. Amy is Gilda Ratner, Tona says. That would be good. Latasha Leone, says John. Sarah Silverman. Julia Roberts. Maybe who plays Leonard from The Big Bang Theory could work. I guess that would be for me, right? So it's Waiting for Godot that Keanu is in. It's interesting. I did Waiting for Godot in college. It was a lot of fun. He still wants to know, do all viruses follow the universal genetic code? I.e., do any use non-standard bases? Well, as you know, that genetic codes are optimized in different organisms, right? So there's a human code because many of the triplets, the codons, are degenerate. So there can be up to 6 codons coding for one amino acid, and so that codon composition is different in different organisms, right? And so the virus usually matches its host in terms of codon usage. And so if you want to make a vaccine of a human virus and you want to grow it in some animal cell, then you have to codon optimize it, right? If you want to produce a spike protein in E. coli, you have to codon optimize it for E. coli. So that's one thing. That's just the genetic code. But you say non-standard bases. There are non-standard bases in viral genomes. Yeah, for sure. Phages in particular do a lot of non-standard. There's a fifth base that can be substituted for a lot of the others, and their modifications. So yes, they certainly exist, non-standard bases in viruses. So you're up to code. Danny says you're about Bruce Lee's height. I would go with a young actor who resembles you early in your career. Then the actor can be made more mature later in the film. It's all very interesting, this idea. Instead of fantasy football, we're going to have fantasy TWIV movie. That's right. So there is a TWIV documentary out that I have a reasonably good role in. Have you seen it? Um, let's see if I can find it on YouTube. Where is it? Gosh, if I search for Racaniello and COVID, I get all kinds of stuff I did during the pandemic. Let's take Chan out. Anyway, I'll give you the link so you can watch it. It can be fun. Darn, I got this the other day by, maybe I should look at my history. Yeah, that'll do it. History. Watch history. I watch too many YouTube videos. Here it is. Unmasking COVID-19's true origin. I'll put the link here in the chat. There you go. So tell me what you think. I mean, they have lab leakers in it, which really bothers me. But otherwise. And Logotic says, Gary Oldman as Daniel Griffin. That would be good. You could have Daniel in it, right? So here we can make it a TWIV movie. So we could have who's going to play Dixon, who's going to play Kathy, Rich Condit, Brienne, Angela. Well, some of these people could play themselves, right? Who would play Dixon? That would be interesting. Is there, Patricia wants to know, is there a new variant that's concerning? There is a new variant, and it's drifted antigenically, and we don't know what that is going to do against the booster that's currently being used. You know, so few people are getting boosters in the U.S. anyway. You know, variant matched boosters. So I don't think it's an issue, frankly. But you know, the T cells are going to protect you against severe diseases. You can still feel crappy, but with these variants, the T cells are still going to work. Does this year's flu shot protect from the circulating strain? Again, we don't know. So there's drift in this, right, this K clade, but what that means clinically is another issue, and we have to collect those data, right? Rita asks us the physicians who got measles were not vaccinated. No, they were vaccinated. That's why they got mild measles, right? Yeah. Patrick Wilson. That's interesting. Yeah, David Tennant. That's a good one. Who's the guy in The Conjuring? I don't know. Maybe somebody else can think of that. Jeff Goldblum. That's interesting. Call me Ishmael for now. I've seen you before. Jeff Goldblum can play just about anybody. Not hard to play me. I have no character, right? Maybe it's hard. It's boring. Nevitz, Gilda Ratner, Rosanna Dana, or Emily Litella. Could have an entire movie of just Jeff Goldblum playing different people. Jeff plays himself. And why Cohen has mixed feelings about the mayor-elect, definitely the best of the options. Mark writes, minus RNA must be converted by an RNA polymerase, a viral RNA polymerase, to a plus sense copy to feed into the ribosome to make viral proteins. Jeremy Davies in Saving Private Ryan. My kid just sang Vincent the Virus Science Guy. How cute. Very nice. Thank him again for his lovely virus drawings. All right, GSD Bio writes, do you think phages displaying poliovirus antigens could be used as a viral vectored OPV, since it might be able to avoid the issues with the current IPV and OPV and keep the benefits? So it's not going to replicate, right? So it's not really an OPV, but it's going to present antigens, so you could, it's not going to reproduce, right? So if you take it orally, whatever the inoculum is going to have to be enough to induce a good immune response. And, OPV is good because it reproduces in the gut. So I don't think so, because it's not replicating. But maybe some other carrier or organism that replicates could do it, right? That would work. Sorry, One Puddles One is Steve from Kauai. Sorry, Steve. Very, very, I'm sorry, I forgot. Yes, it was up earlier today. Yes, for any of these RNA or DNA, some viruses are challenging for vaccine development. It's a complicated topic. It's not restricted to RNA or DNA. Nevitz writes, talking about phosphates, how could of all scientists have thought the DNA was phosphates on the inside and the bases oblong on the outside? Coulombic repulsion would be massive. And that was Linus Pauling. I don't know what they were thinking. I really don't know. It's a good question. But one of the prevailing theories in 1950s was it's a triple helix, 3 strands, and I don't know where they got that from. It's a good point. Patricia wishes us all happy holidays, filled with faith, hope, joy, and love. Thank you, Patricia. Melinda, that's why I asked you, because I listen to a lot of talks and there are things I hear that I doubt. Good. No, you should doubt. You should be suspicious. Even if it's a virologist saying something, you should, if it sounds funny, not truthful, you should be suspicious, right? Now, I am not going to tell you things purposefully wrong. That's disinformation. Sometimes I get things wrong, right? So I had Babesia on a list of bacteria last week. I know it's not a bacteria, but I just made a mistake. And I got called out, and I said, yeah, you're right. So, yeah, there's no difference in DNA and RNA vaccines. Yeah. Plastella says, I'm probably going to go digital blackout the last week of November. I can't deal with holidays anymore. Okay, we'll miss you. We'll miss you, Questella. I think you contribute a lot to this live stream. Okay, so we will miss you, and we'll see you in December. Peter Sellers from Tim Jones. It could be, yeah. It could be. Nevitz says, Vincent, all press is if it bleeds, it leads, or how scared can we make the public? Yeah, that's absolutely true. Media sensationalism. There you go. Mark, all the media cares about is sensationalism. That's right. Costello wants to get Anthony Hopkins in this movie. Let's see. Who could he play? So it's a kind of a devious person, right? Well, there could be other people that are, peripherally related to TWIV. And he could play Richard E. Bright, right? The lab leaker from Rutgers. It was very devious. Oh my gosh. John wants to know, did they try combining antivirals for COVID like the HIV/AIDS? No, actually, there were some experimentals, but nothing ever got into people. And that's because there's very little resistance to remdesivir and Paxlovid and molnupiravir. So it's really interesting that there's very little resistance. John Wick is another one. who's going to play Dr. Griffin? Well, we had a suggestion for, who was it? Damn. I go back. Who did they suggest for Dr. Griffin? Who did you guys suggest for Dr. Griffin? Gary Oldman. There you go. Gary Oldman. Okay, I'm going to pause and just show you some pictures from Toronto. So I went to the ASTMH meeting with Daniel and Christina from TWIV, and also Chuck Knersch, who's on the board of Parasites Without Borders, and then two women who manned the booth. So Daniel purchased a booth. So at these meetings, there's an exhibit floor where companies set up booths. You rent the booths from the meeting organizers, right? You pay money for it. And you set up booths and people come. And they had a Parasites Without Borders MicrobeTV booth, right? It was just jammed because they're giving away stuff, giving away books and stickers. And they have a quiz, identify this parasite. They have a microscope. You look through it and identify it. So people are very excited about that kind of giving away education, which is our philosophy of the two companies, right? Parasites Without Border and MicrobeTV. We want to educate people for free. So we did a TWIP there. We did a TWIV clinical update. So I have some pictures. Let me show you. Because Kara, who is one of the ladies who ran the booth, got a photographer for a day. So this is what the booth looks like, right? It's got a table in front. It's got some of these banners. It's got a big thing in the back, Parasites Without Borders, and you take pictures in front of it. And, you can see we're giving stuff away. And people are coming. They want to look. So let's see some of these pictures here. So here's a bunch of pictures of us during TWIP. Right? So I don't have to tell you who's who, but these are sharp. Daniel looks very sharp. Yeah. I look ridiculous here. And this one on the left is ridiculous. A little better. I guess my facial expressions are weird. Oh boy, I have to moderate myself. Christina looks very nice, right? So here we are, TWIP. You can click on it and make it bigger. Sorry. Martin Grobusch was our guest. And, I set up all this recording. This is what I'm talking about, schlepping equipment, right? So I got this tripod, I got this camera and lens, which is really heavy. I got all these microphones, which are pretty heavy. They have a very heavy metal thing here that connects to the XLR cable, which is heavy. Then I got to bring extension cords. And so I'm just trying to cut that down. So there's Daniel. And we can just go through these here. It's me. It's not so bad. 4 of us. Daniel. Martin, who was very good. Christina. I like these candid shots that he took. And there's the setup. So we were just out in the hallway of this convention. It was pretty noisy, but the final, which comes out Friday, is actually pretty good. And these are the kind of banners that are at the booth, but we moved them up so people would see while we're doing the podcast, get some attention, right? And this was the little area that they gave us. It was just kind of walled off with a cloth there, and TWIP started at 7:30, so there weren't a lot of people there. But TWIV clinical updates started at 9, and there were far more people there. Martin Grobusch, an interesting infectious disease doctor. Another banner for TWIV. This is still TWIP. Martin was very animated. He was good. I didn't know what to expect at all. TWIP, still TWIP. Okay, so that was the space they gave us. We got these banners on either side. There are tables in the front and then chairs set up in the back. And people are walking around going to session, so that's why it's a little noisy there. That's a nice shot. I like that. You can see Daniel's Parasites Without Borders socks. Yeah, Daniel looks good. Martin Grobisch. Yeah. You know, when we're all together, it's different because we're seeing each other and we're reacting to each other in a different way. And there's the crew, the 4 of us. Some headshots. I like this one. This is fun. It's a picture of a phone focusing, and they got the little focus box around my head. That's cute. I like that. Daniel likes it too. So this is clinical update, me and Daniel. We had sitting on 2 nice chairs there, 2 nice cushy chairs. And we're using our laptops because that's how we reference all the papers that he's doing. We had more people for TWIV because it was later, as you can see. A lot of fans. Is really grateful. This is good. I like that. Very nice shot. This is a hand. Daniel does that with his hand often. Yeah. And that too. He does that kind of motion with his hand. And I like me in the background there. That's cool. Oh, I mean, you got to get the creepy guy to play me. I mean, look at that. Oh my God, I look so creepy in that shot. Oy, oy, oy. And some audience shots. Yeah, we had a good crowd. I mean, not many more seats left, right? I like these audience shots. They're good photographers. So nice, cushy white seats. Knockoffs of whatever that original was. Okay, so this is the booth now, right? And people come up and interact. And then you can go back and sit in here. There are chairs. There are bookshelves with the books on that they gave. They gave away 8th edition of parasitology, parasitic diseases. And I sat here a lot and people would come up and chat with me. So here's some of the stuff we're giving away. Also, gummy worms, yuck. And here, different life cycles, and you had to guess which one was which. I like that. I think that's really good. QR code for a newsletter, and, you know, just the sense of people walking up and being interested in a book and getting one. Yeah, it's a nice booth, see? I like to sit in those chairs. They're very comfortable. And they had a TV monitor there playing a loop of a video that had been made, promoting all these activities of Parasites Without Border, right? Textbook certification exams, MicrobeTV, and then all other things on the website. There's Kara, who runs a publicity firm in Florida, and she takes care of the booth every year. People were very happy to get a free book, even though they had to carry it. Okay, let's see. Yeah, you see there are a lot of booths around this floor here. I think this one did really well. Which I think you can get a sense of. There are a lot of interested people, right? Looking through the microscope to identify parasites. And Christina spent some time at the booth as well, talking to people. And a couple of pictures. People wanted pictures with us. I'm sitting there chatting with Daniel. Signing a book for somebody. That's me. I like signing books for people. It's fun. And we had a picture of Dixon there at the booth in honor. This is called Despommiers parasitic diseases now, from now on, the 8th edition and forward. Despommiers parasitic diseases. We gave away like 100 copies of them. There you go. And this is a reduvid bug, a big blow up of a reduvid bug. I love that. Me and Daniel, the vector of Chagas disease, and me and Daniel. That's a good picture of me. I like that. Daniel always looks good. And here's the crew, right? So that's Kara, who's running the booth. And I forgot this young lady's name. She works for Kara. Christina, Daniel, and myself. And a horrible picture of me. Okay. Okay. So that's photos. And we'll go back to some comments before we do our mini lecture. Let's get back to... So Melinda wants to know, why do some viruses temporarily increase the risk of heart attack and stroke? So I think there are two main components. One is cytokines and inflammation, right? There is a lot of production of cytokines that are damaging to various tissues. And that's also responsible for brain effects, right? Cytokines get into the brain. So I think they cause stroke and cardiovascular issues. But also, when you have severe lung compromise, the heart has to work very hard, right, in order to provide enough oxygen to tissues. And so this puts extreme stress on the cardiovascular system, and that can lead to heart attack as well. And that happens with many respiratory viruses, right? As you say, viruses in general. Yeah. Nevitz says, I also have the Ed Norton hang up. Jackie Gleason would yell, Norton, in all of the episodes, Honeymooners episodes, imprinting Ed Norton, methylation on the brain to an entire generation. That's right. That's right. Tina Fey can play anyone, including Amy. Yeah, she would be good. When Amy said, "You think?" Definitely Joan Rivers. That would be great. Can't forget Dixon. I haven't heard a suggestion for Dixon. John likes Keanu Reeves in Sweet November with Charlize Theron. I didn't see it. Vista says, pretty sure I had H3N2 in 1970. Well, it's still circulating, right? It's very likely you still got infected. Will I be resistant? How much can H3 change and still be H3? Well, it can change a lot, but, you know, we don't get new H's. We have H1 through H... How many do we have? 18? 19? We don't get new ones arising. We have some that we didn't know. So 18 and 19 were discovered in bats, I think in Guatemala. But, you know, H3 doesn't become something else. You know, you get 1, 2 amino acid changes, and that doesn't change whether it's an H3 or not. Now, would you be... resistance is not the right word, right? Because you're always going to get infected. But whether you get severe disease or not is the question. And I think, you know, you're going to have a T-cell response that's going to help prevent severe disease. But it's more likely that you've been subsequently infected with H3N2 because it's been circulating ever since 1968 till now. It's still circulating. You know, it trades off with H1N1. Some years H3 is higher, some years H1 is higher. So I'm thinking you're getting boosted. If this variant doesn't match the vaccine well, it could cause more severe disease. But in the end, you won't die, as Daniel says. You still will be protected against really severe disease and death. Anthony Hopkins would be great to play Dixon. I think that would be good. Remember, Dixon is a very jovial guy, right? I'm not getting a jovial vibe from Anthony Hopkins. You know, I'm getting a creepy vibe from him. So I don't know about that. But he's an actor, right? He can play different people. Do you think the U.S. will lose? I don't know what you're talking about. H5N5 is from reassortment of high and low path. I think so, Jonathan. I think so. The N5 from the low path, but the H5 from the high. Logotic is not surprised that I'm a Samuel Beckett fan. When did I say that? Are you talking about the Irish playwright? I don't know. Tell me what you're thinking. For Amy, Jordan Jensen has the same moxie. Melinda's more concerned about flu season because they declared an epidemic in Japan. Okay, well, there are epidemics every winter of flu in temperate climates, right? So we have epidemics in the U.S., and it's starting to build up in the U.S. already, right? Do I think the U.S. will lose its measles elimination status? Yes, in January it is going to have 12 months of unchecked circulation, so that does it. So we lose it in January. When do you think we'll start seeing cases of polio paralysis? Polio vaccination rates are still high, except in pockets. So this is the thing. There are always pockets of low vaccination. For example, in Rockland County, New York, very low immunization rates, and that's where we had a single case of polio in the summer of 2022. So, and that's before RFK Jr. and his ilk came in, right? So, I mean, there have been some predictions, some modelings done, and we covered this on TWIV, actually. In 10, 50 years, there could be tens of thousands of cases. So right now, I don't see resistance to polio vaccination. So I don't think it's imminent. But who knows what's going to happen? It could be, I don't think it's going to be next year. I think 5 years, within the next 5 years, we might see some polio cases if immunization rates drop. So far, I haven't seen that. Anthony Hopkins as Dixon would warm Questella's heart. SRR is donating through YouTube to raise the algorithm. Thank you, SRR from Paso Robles. Questella, thank you for your support of science communication. Really appreciate it. Tona wants to know, what if all the TWIV team played each other and not limited by gender? Who would I be? Oh gosh, I'd love to be a lot of TWIV team members, not limited by gender. I could play Angela Mingarelli, but I'd have to work out for a while, I have to get in better shape. But that would be fun to play, because I know their personalities, right? I see them well. And so do you guys if you watch a lot, right? That's very funny. That's a good question. Not uracil, but there are modifications of bases that would occur, right? So not just human versus viral uracil, but it's a different base altogether. If you want, we can do a mini lecture on base modifications, okay? So we can get it all correct. SRR, thank you for your contribution to science communication. Really appreciate it. Keanu Reeves to play Daniel. That's a good one. Nevitz says, Bruce Lee as Vincent in The Way of the Virus, coming to theater soon. That's really good. I love it. The Way of the Virus. Brilliant. Brian Cox could. Dixon, I guess, right? Anthony Hopkins. Brian Cox again. Vincent with the Muppets. Follow that virus. And Tractor Poodle is in Bangkok. Welcome. We have another person in Thailand at the moment. Very cool. So we got Vincent, Daniel, Dixon, Alan, Kathy, Brianna, Angela, Rich, Paul, Paul Offit. When I'm thinking of playing someone else, I'm trying to find someone that's far from me. I mean, I could easily play Daniel, but that's not a challenge. Or I could do Rich easily, right? I could even do Kathy Spindler easily, but Angela is much younger person, right? 50 some years, almost 50 years younger, and in good shape, and has a certain way of talking, and I'd love to be able to do that. Yeah. David Attenborough for Dixon. That would be cool. Nobody's picked Bill Nye for anyone. That would be interesting, wouldn't it? Ron Coleman, Aerosmith, 1931. Wow, that's something. And this is interesting. Lagotic says, Dixon always reminded me of John Huston. John Huston would be a great Dixon, yes. He even kind of looks for him. Amy Irving for Amy. Oh my gosh, Questella. The end credits will be Vincent playing the guitar for all of us finally. You remember? I thought everyone had forgotten that. Oh my gosh. So what Questella is referencing is sometime during the pandemic, early on, people saw the guitar. You can't see it anymore. It's back here. No, I can't show it to you. I'm wired up. And someone said, do you play it? I said, I haven't played it in a while. And they said, well, will you play it when the pandemic's over? And I said, sure. And I figured out everyone forgot about it. But Questella, I'm very impressed. You have a great memory. Who plays Mark Martin? Oh my gosh. I don't know. I'm not a good. Yeah, and Tona, same thing. Someone funny. So I think Robin Williams would play Mark Martin if he were around, right? He would be really good. Oh, you still come to office hours? Thank you. Thank you so much. If you want a break from social media, I totally understand. But if you come here, we'd love it because we'd miss you. You're always here. You're always vocal. I love it. And you, in the beginning of office hours, when there was Q&A with A&V, you said, this is going to last. It's going to work. And you were right. Jonathan, good point regarding the hypothetical phage OPV can replicate in the gut by infecting a probiotic bacterial strain. Right, a safe biomimicry of an actual enteric infection. Very good. Now, so that now could work. Right, you just have to make sure it doesn't alter your microbiome, right? So maybe it can be, well, it can't be lysogenic because then you won't get new phage produced because it's the replication that we want. But you're saying a probiotic strain, so maybe you give the strain like the day before you give the vaccine. Interesting. It's an interesting idea. Sandy, I listen to a lot of podcasts and don't chat live lately, but I always like being here. I see you every week. You always say at least something. Anthony Hopkins does a lot of drama too. Rip Taylor for Mark Martin. Vincent, have you fooled around with Sora, the AI? I have not. I have fooled around a lot with AI, Google AI for images. It's called Nano Banana. And in fact, this picture that I showed you of me where I hate with my hair's too long. And I told AI to make it short. And it did a great job of cutting my hair. Oh my gosh. I should show it to you, but I don't have the picture here. Anyway, I haven't played around with Sora. Brent Spiner for Dixon. Claire Danes for Angela Rasmussen. Well, yeah, she's TWIV adjacent, right? So that could be. She could be part of it. Will says, Marty Makari lies about clinical studies. It's important to choose which expert to follow. So, yes, when I was talking with Melinda before, I was trying to basically imply that, because not all experts you should listen to. And how do you know, right? So you have to establish a rapport with someone. Like, you've been hearing me for years, right? And my team, all of the people on my team, you've been hearing us for years. You can trust us that we will give you accurate scientific information. And, Marty Makari's an MD with no particular expertise in virology and infectious diseases. And he lies all the time. That's the thing. And you know this, because when he says something, there are plenty of analyses left and right, including Paul Offit and us. So, yes, it's important to choose what expert to follow. Questella, this movie is going to cost a lot, because these people are all famous, right? But, maybe it could be a movie about science communication, and they'll donate their time because it's a good cause. Yes, we need to talk all these lists into doing it for science communication and recognition. That's right. It's a good idea. Caulfield wants to know, Caulfield, are viruses still becoming incorporated into genes? Most likely. So viruses can integrate into DNA, right? They can do it as an obligate part of the reproductive cycle, like retroviruses, where there can be an accident, just pieces of DNA being incorporated. And it can go into a somatic cell, which is most of our cells are somatic cells. Then we have germ cells, right? Sperm or eggs or ova. If they get into somatic cells and integrate, then somatic cells turn over, except for neurons, they turn over. They die, so it doesn't have much consequence. If they get into a germ line, it can be passed on. The point is, we're not going to see this, because, if somebody in Mongolia gets an integration in their germline, if that happens to be propagated, we wouldn't see it in enough people for 10,000 years probably before we started saying, oh, and we'd have to look in a genome. We'd have to look in a genome, which, we're doing more and more. We're sequencing people's genomes, so presumably we would pick it up. But yes, they are still being incorporated, but it's very hard to see it for the reasons I've told you. Yeah. Dorothy from Duluth, with the temperatures flirting with 0C, about the only conversion I know instantly. And I even took enough science courses. I'm somewhat embarrassed by that. Oh, yes. So 0 is minus 32, right? But we also have, there's some other instant conversion. 28 is 82. That's easy, because you can flip it around. And 20 is 68, of course. So those are easy conversions. There are a couple of others, too. Pete Glass writes, I found something amusing in my late mom's family documents dated 17th February 1966 from the local medical officer of my school. BCG vaccination certificate. But the smallpox cert was lost with a stolen passport. That's very cool to find your old vaccination records. Questella, need to include John Delancey while we're at it? Sure. James Cameron. Well, we're going to have a director, right? Tono has a question about an HPV shot for a young adult male not yet sexually active. Tell me 3 reasons why it's in his best interest to get HPV vaccine. Penile cancer, anal cancer, oral cancer. He can get all 3. Horrible cancers. And you want to vaccinate before becoming sexually active. That's the key, right? Although that study that we did on TWIV says even if you get infected, the vaccine has a 75% chance of clearing the infection, which is really interesting. But it's it's 100% protecting you against cancer when you get it before you become sexually active. All right, so those cancers are terrible, and men can get it from other men, or men can get it from women, and women can get it from other women, and women can get it from men. So that's why, those three reasons. Meryl Streep would be awesome as Christina. That would be funny. Yes, so video with a smartphone. Don't forget to take a battery backup. So I've done some tests, right? And, a live event is typically not more than an hour, and a fully charged phone, no problem doing 4K video for an hour. I also have plenty of storage because I bought extra storage, right? So, but yes, I charge it up. But the thing is, Will, the microphone, the wireless mic plugs into the only port on the iPhone anyway. And so it has to be fully charged before I start that. Yeah. Tona watched the trip on YouTube, and you and Daniel looked awesome and sharp in your black and white and gray. Thank you, Tona. Daniel looks to be an actor in another life. Yeah, it kind of looks like an airport, right? I wonder if we did a podcast in an airport, nobody would pay attention. Everybody's so focused on getting to their plane, or they're watching the bloody sports, which are everywhere and you can't escape. Oh my gosh. John donated through Parasites Without Borders this week because Dr. Daniel said they're matching up to $20K to MicrobeTV. Thank you, John, for mentioning that. So for November, December, and January, if you donate to parasiteswithoutborders.com, you can just go there, and there's a big button that says donate. They will match your donation, and at the end, they're going to give up to $20,000 to MicrobeTV, and we would love that. So if you're going to donate for these months, it's good to donate to Dan. But remember, Daniel only goes up to $20K. Anything above that, he's going to keep for his Parasites Without Borders, which is fine. But I'm just saying, not everybody has to go there. Yes, gummy worms at the parasite table. That's cool. Yeah, I didn't come home sick. In all of my travels this year, I was fine, and I'm masking on planes and public transport and so forth. So thank you very much. Don't cry. We love all of you. Yes, Tona, I saw your email today. We'll give it to you December 22nd in person. We have to get Daniel and, Daniel to sign it because there's nobody else to sign it, right? Yes, isn't it a nice way to honor Dixon to make it Despommiers' parasitic diseases? He would love that, right? He would totally love it. Triple-stranded DNA was Pauling. Had he heard of Chargaff's rules? I don't know if he was believed. That's the thing. Rob Reiner is Dixon. Anthony Hopkins is a serious actor. He doesn't just do Lecter. I understand that. He's a serious, but that's what's in my head, right? Angela Mingarelli could play me. Oh my gosh, this is so funny. Vincent, do you start teaching again in January? I do. And the lectures will go up on YouTube as usual. And that reminds me, I got to put the syllabus up because registration started and the class is already full, 120 students. And people are emailing me, can I take your course? It's my last year. Rainey. So Rainey is the name of a major character in Peripheral and Agency by William Gibson, 2 books that I just finished. And that reminds me, I wanted to tell you, I had read Peripheral completely. I had little clue what was going on. So I just reread it, and it is 100%, 99% making sense when I read it the second time. Everything made sense, and I read more slowly, and I could reference. It was great. So I think someone had suggested to read it again. It worked. Thank you very much. Anyway, Rainey, you are a character in those books. I want to do a mini lecture now, okay? Because it's not going to take too long, and I think it's cool. So let's do that. So this is a little bit on gene editing. You know, I'm not an expert on gene editing, but I understand some things. So there have been some CRISPR-based gene editing solutions approved, and one of them is for blood diseases. So here's a Nature article from November 2023. The UK first to approve CRISPR treatment for thalassemia and sickle cell disease. So these are diseases of hemoglobin, right? So in the thalassemias, you can have low or no hemoglobin, and in sickle cell, mutations that cause red blood cells to make a sickle form, and it's, you know, blood flow is affected, they clump up and so forth. And so these are monogenic diseases that are amenable to correction. And so this uses CRISPR. And this is a very clever thing that they did for this beta thalassemia. So here's a Nature Medicine paper from 2022. CRISPR-Cas9 mediated gene editing of the BCL11A enhancer for pediatric beta-naught transfusion-dependent beta thalassemia. So these are babies that are born with mutations in both copies of the hemoglobin gene. So they're dependent on transfusions their whole life, right? You have to periodically get transfusions to make enough hemoglobin. And so in this paper, what they did is they edited the DNA to turn on the fetal hemoglobin. So BCL11A is an enhancer for fetal hemoglobin, and they found that if you mutate it, you can turn on fetal hemoglobin, and that'll work for the life of the individual because it substitutes for the mutated hemoglobin. So here's the explanation. In some patients with thalassemia, continuous expression of fetal hemoglobin, right, HbF, so this is usually turned off, and it's turned off at the level of the enhancer. The enhancer is a sequence away from the transcription start site of the gene that can enhance transcription. And so at a certain point, the enhancer is turned off and you don't make any more fetal hemoglobin, right? But they found that some people have continuous expression to adulthood, and this can alleviate the symptoms of anemia. So that suggested that you could use fetal hemoglobin to replace the functionally impaired hemoglobin A as a treatment for beta thalassemia, all right? And so then in separate studies, in genome-wide studies and animal experiments, they found that the BCL11A enhancer negatively regulates the expression of fetal globin, right? So what they do is they make a CRISPR Cas9 guide RNA, and they disrupt BCL11A. Right? They disrupt the enhancer, and it induces fetal globin, and therefore they say this is a practical therapeutic strategy for treating beta thalassemia and sickle cell disease. So you just mutate the BCL11A enhancer, you turn on fetal hemoglobin, and now they're making their own hemoglobin, and it works. It's a totally perfect substitute for adult hemoglobin, all right? So that's what has been done, and in fact, this has been licensed. So how does it work? Well, in general, this is how CRISPR-Cas9 gene editing works. So you have your DNA, right, your chromosomes, human chromosomes here, and these are all coiled up DNA. So here we've uncoiled a little bit of our DNA. And let's say we want to make a change in the DNA of a cell. So first we design a guide RNA. So half of the guide RNA is complementary to the sequence in the DNA that you want to change. Okay, it's called the guide RNA. And then the rest of the guide RNA allows it to bind the nuclease Cas9, which is part of the CRISPR system. So you can make the guide RNA and the Cas9 protein from the same vector, right? You can deliver it to people. And so they will make the guide RNA. It will associate with Cas9. Then that guide RNA will hybridize to wherever the sequence is in the DNA. And then the Cas9 will make a double-stranded cut. If I get two scissors here, double-stranded cut. And now you have a double-stranded break. Now, the cell does not like double-stranded breaks. It will repair them immediately. And so one thing is that you let the self repair the cut, and it usually will join, take some bases off and join the ends. And this is called error-prone repair because it's not maintaining the reading frame. So the protein that's encoded here is going to be disrupted. But you can also edit a gene by providing a piece of DNA that overlaps with the cut. So you, along with the guide RNA and the Cas9, you also provide a piece of DNA. It's shown here in green. That is overlapping the cut site and has the corrections that you want to make. And that is recombined. That is used by cellular enzymes to repair the break. It substitutes in the inserted DNA, and now the DNA has the changes that you want in it. So that could be to alter the enhancer for fetal globin synthesis, for example. So that's CRISPR-Cas9 based gene editing. So you could use this to edit anything, whatever you want. You could remove CCR5, which is a co-receptor for HIV, and make people resistant to HIV. And that's, in fact, what the Chinese scientist did a number of years ago, and he did it without any permission. He went to jail and big brouhaha, right? Because we're not ready to do germline editing. When we do it on globin, it's not germline. It's usually you use stem cells. You can edit the stem cells, and then they give rise to blood cells that have been edited. But that child will not pass those modifications on to his or her offspring because it's not in the germline. Now, the problem with CRISPR-Cas9 is that it has off-target effects. So this guide RNA, yeah, it will hybridize to the sequence you want it to, but often it will hybridize somewhere else and cut somewhere else, and that can cause problems, right? You don't want it cutting in other places because you don't know what the effect is going to be. So this is why we're not going to use CRISPR-Cas9 for somatics, for germline editing, because it makes too many off-target effects. And in fact, we shouldn't even use it for stem cells because then you can make a lot of stem cells that are mutated. So what's the alternative? So here's one example. This is a recent example. This is in May. The world's first personalized CRISPR therapy given to a baby with genetic disease. So this baby had inherited mutations in the gene encoding carbamoyl phosphate synthetase, which is involved in the processing of nitrogen-containing compounds when the body breaks down protein. Okay, so as a result, you have high nitrogen, high ammonia in the blood, and that's toxic to the brain. So this is not good. They decided to use CRISPR to treat, to change the gene, so it was no longer mutated. And it was obviously personalized for the baby. And this was done by base editing, which doesn't do a double-strand break. It does a single-strand break. So remember, CRISPR, you have a guide RNA that binds your RNA to the Cas9 enzyme. Then you have your target sequence that finds the part of your DNA that you want to edit. The Cas9 makes a double-stranded DNA cut, and then you can let the cell do non-homologous end joining, as I said, or you can add a donor DNA and have it patched in there and replace the sequence that's been removed, right? So that's a double-strand break, and this is problematic because if this guide RNA hybridizes somewhere else, you're going to make double-strand breaks in other genes. But base editing converts one base to another without creating a double-stranded DNA break. And so what happens here, you have the same system, but this is a dead Cas9. It's been altered so it can only make a nick. It doesn't make a double-strand break. You have a guide RNA that brings the Cas9 to your gene, but you also have fused to Cas9 a deaminase in this case. You could fuse any base-modifying enzyme, but here's a deaminase. So the Cas9 will make a nick, but it will also change this A to an I, for example. And then after DNA repair, it templates as a C, and you end up putting in a G, and then the other strand has a C. So you make a CG change, one base only. So if you have a single base mutation in a gene, as with this baby, you can correct it this way. So that's the key here, is that you don't make a double-stranded break, and that makes fewer off-target effects. So this was done in people a couple of years ago to lower cholesterol. So they used it to switch off a gene that regulates bad cholesterol, right? The bad cholesterol. But safety concerns, if a person died in this trial, and it's not clear why yet, so it's not good, right? So we're going to have to figure that out. Then there's also prime editing. So these were the two alternatives to CRISPR that were mentioned in the article that I read 2 weeks ago. This is prime editing. This is way more complicated. And from my eye, it seems to me it's too complicated. And something's going to go wrong, okay? You have the same idea. You have a Cas9 nuclease, but now it's fused to reverse transcriptase. So that's the blob up here, okay? So you got Cas9 nuclease fused to reverse transcriptase. Again, the nuclease is modified so it only makes a single-stranded DNA nick. So the guide RNA is different also. It has a part of the RNA is going to be directing this complex to the part of the DNA that you want, and then it has a hairpin and an additional sequence that is going to be edited right up here. So this is happening on both strands. The Cas9 will nick the DNA, and then this this primer essentially at the top here is going to hybridize to that strand of DNA after the nick. So after the nick, that strand is going to be released. It's going to hybridize to that DNA, and that's going to serve as a template for reverse transcription into DNA, because here we have an RNA, so that's RT template there. And then you're going to end up with an insertion or a correction. And this makes a little flap here, so that's shown down at the bottom here. You can have a piece of extra DNA at the 3' end or at the 5' end, and then these get inserted into the strand to correct it, to correct the defect that you want to. So it depends on the activity of reverse transcriptase here to copy the RNA into DNA and then get that repaired into the DNA. So this, again, is a single-stranded break. In theory, has fewer off-target effects. So those are the two alternatives to CRISPR-Cas9. This is base editing and prime editing. So they do single-stranded breaks and have different modalities. And this last one, prime editing, has been trialed in a person. This is a Nature article from May. This patient had a mutation in a gene encoding a subunit of NADPH oxidase that ends up causing a disease called chronic granulatomas disease, which affects a variety of immune cells, including neutrophils. And this was a teenager. He got therapy here. He got the prime editing therapy. And a month later, it seemed to restore the function of this enzyme in 2/3 of his neutrophils, which would have been enough to boost his immune system. So this case, it worked, and a single individual, and he was fine with no serious after effects. So these are obviously all experimental and have to be developed some more, but just so you understand what is going on here. Now, how does this actually work? Well, there are two modalities, right? So you can deliver the CRISPR-Cas9 or the base edit or the prime edit system right in a virus to the patient if you want. And so in some of these studies, that's what they do, right? They just deliver the virus. It goes to many cells and corrects the defect. And the more you do it, the more correction you're going to get. Or you can take out cells from the patient, put in the gene editing solution, and then put the cells back into the patient. So, for example, if you take out hematopoietic stem cells and you have a globin defect, you can correct the globin defect in cells in culture and then infuse those back into the patient, and they will give rise to blood cells. All the cells in the blood come from the hematopoietic stem cells. So now you've repaired the gene and you put it back in the patient, and it will the cells will start making lymphocytes and red blood cells with the correction. This is how CAR T cell therapy works. They take T cells from the patient, they infuse they infect them with a virus that makes this chimeric antigen receptor that now directs the T cells to a tumor antigen to kill the tumor. So those are taken out, they're infected in vitro and put back into the patient. So those are the two ways. You can give the patient a virus or you can give the patient virus-infected cells. So these are all still early days and need to develop more. But it's amazing that we can do this, and it's progressed a lot in the last 10 years. And, that's basic research. Nobody knew that CRISPR-Cas9 was going to be useful for gene editing. They just stumbled on it. And then when people saw what it could do, we now have gene editing. And now these modifications are basically using components of the CRISPR system. So I think it's really cool. And we need to support basic research in this country, folks. Can't be cutting left and right. Okay. Robert De Niro for Dixon. That could be two. Mark wants a roach to play RFK. JNNJ, thank you for all your teaching and expertise. I use your lecture slides in my grand rounds. Would love to have you talk to our doctors here in our little island of Saipan. I would love to combine it with with one of my travels in the future. You can email me, Vincent at MicrobeTV. Yeah, Bill Nye is not an actor. Yeah, I know, but he could probably act, right? He could just be Paul Offit. Bill Nye could be Rich Condit. Yeah, Bill Nye could do Daniel. Yeah, John remembers the guitar. Oh my gosh. Yes, Vanity Robin Williams playing Mark Martin, right? Yeah, Tom Hanks is versatile. Bill Nighy is an actor. Robert De Niro would be a brilliant Dixon. Jack Black. Who's he going to play? Vincent and I were going to sing my parody. SARS-CoV-2 is coming to town to the tune of Santa Claus is coming to town. Hey, why don't we record that, Vanity? Yeah, we could do it online. That would be fun for the holidays. I don't sing very well. But, yeah, Kathy and Rich can sing. If you want to record with them, that's probably better than me. Lytic lactococcus it's what I work with. Okay, so you're doing it. That's cool. Should we ask ChatGPT who should play you? Sure, let's see what they come up with. Yeah. Who would play Nels Elde? I think that guy who you suggested for me, whose name I forgot now. Let's see. What's his name? The guy who was in Broadchurch. I can't remember. David Tennant. Yeah, because he's kind of tall like Nick Nels is. Gary Sinise would be good to play Alan. Yeah, we haven't talked much about Alan. Up to Code Danny says, I know someone who has had COVID at least twice. They're not vaccinated. What level of immunity could they have by now? They have some immunity, but it would be best to combine it with a vaccination to get the optimal immunity. So a combination of infection and vaccination is really good, but we don't tell people to do that because you could get very sick from getting infected, right? So it's not a good plan. But you do need a vaccination to complement that. This is an interesting question. How is the issue of AI to create new viruses? Is that an issue of creating a lethal virus? So AI doesn't know how to make a virus because no one has made a lethal virus, right? And so AI doesn't know how to do it because it's based on what it gets. So it would have no clue. And I think if you asked it, it's probably got some checks in there to say, sorry, I can't answer that, right? You try and make certain pictures, they won't do it for you, right? So I'm sure that it's built into the system, although you could imagine that some countries wouldn't have it built in or some terrorists would take it out, right? But I don't think it could do it. What would you do? I mean, I know I could do it. I could have some ideas of how to do it, but there's nothing that's published because no one has tried those experiments. But sure, I have ideas. Not going to ever do it. Kevin Costner. Barb tries. See, now I remember. Welcome from Durham. Now, that's all I'm thinking of. Who can play you all? Well, it's a fun idea, right? And this is how office hours works. We keep our focus on the science. I answer your questions. I do a mini lecture, but then there's an underlying thread always of something else that just happened to have popped up, and it's different every week. And this week, it's who's going to play the TWIV people. It's all interesting. So Visto asks, could HIV become a HERV? No, because it does not infect germ cells, right? It has to infect sperm or ovum. Doesn't. Thank goodness. Just infects CD4 positive T cells and macrophages, not germ cells. Who could Clint Eastwood play? I think he could do Dixon, just because he's tall and lanky, right? I think he could do Dixon. He could do Rich Condit, too. Lise, hello, welcome in Columbus 55C. Got Novavax and Zoster, right? That's what you mean by ZR. Has TWIV covered the study on EBV and lupus? Friday. We're doing it Friday. That's in 2 days. It'll be out Sunday. See, this is it. Some MDs won't prescribe Paxlovid. It's disappointing because you hear Daniel every week extolling the virtues of Paxlovid. So I don't understand why physicians don't do that. Oh, it's not serious enough. How do you know? And by the time it's serious enough, it's too late. That's crazy. Oh, sorry, you had zero reaction from Novavax. Very good. A younger Paul Offit, a younger Clint Eastwood could play Paul Offit. Males should get HPV vax to protect partners, male or female partners, okay? And themselves. Works all ways. And that's correct, Melinda. Even if someone's not sexually active, assault could happen, right? Yes. Are other people masking on the plane? one or two. So I'm on a plane with 300 people, right? That was to L.A. And nope, I didn't see anybody. One or two. And then the plane to Toronto was tiny. I don't know, 50 passengers, nobody but me. So I went to L.A. On the weekend to attend the memorial ceremony for David Baltimore. David Baltimore, 1975 Nobel laureate for discovering, along with Howard Temin, reverse transcriptase. He was my postdoctoral mentor from 1979 to 1982. I was at MIT. It was a very nice ceremony on Saturday at Caltech on campus. Pouring rain. Oh my God, I got soaking wet. Friday night, I get into Lax. The Uber pickup, you have to walk to. It's like a mile away. So I walk and I get wet. And then they have these covered areas, but the wind is just whipping and everybody is totally soaked. Fortunately, there was a hairdryer in my room and I used it to dry my clothes. Anyway, so I just pulled this off my shelf ahead of the curve by Shane Crotty. It's a biography he wrote many years ago of David Baltimore. I think I'm going to read it again. But there's a cool part in this book. I don't know if anyone's read it, but on page, let's see, here it is, page 133, it's called an interlude. And what follows that chapter, what it is, it's the genome sequence of poliovirus, which I did, and because I did it in David's lab. And so it's called Interlude, and it says, this is the sequence, all the information necessary to create poliovirus. He tells, once the Cambridge ban on recombinant DNA was lifted, one of the first pieces of DNA that the Baltimore lab cloned was the poliovirus genome. This work was done by Vincent Racaniello, a postdoctoral fellow in the lab. Since polio is an RNA virus, Racaniello first made a DNA copy of the RNA using reverse transcriptase and then pasted it into a plasmid. But there is one thing wrong about this chapter. He says, I did the sequence by Sanger sequencing. Actually, I did not. I did it by Maximin Gilbert. And this was published in 1981. So he could have looked at the paper and got it right. But anyway, it's cool. He's got the whole sequence there in a couple of pages. So I feel honored that I'm in a biography of David Baltimore. Anyway, a little diversion there. Vista says in Australia they did not HPV vaccinate males. Short-sighted cost-cutting. Not good. In the U.S., they realized that initially they didn't vaccinate males either. And then the studies were done and showed that that really makes an impact on HPV in females. But of course, it also makes an impact on men who have sex with men. It's just everybody should get vaccinated. Because you just don't know. You may think you're in a monogamous relationship, but you may not be. I mean, your partner may be cheating on you. I hate to say it, but then, yeah, you guys understand. Katharine Hepburn as Amy. Oh, that's interesting. Yes, I will be recording the classes and making them available. So now Johnson wants to know, what's the movie subject, this movie that we're talking about? Will it be fiction? The never-ending study where Vincent enters a paper and has to battle the growing ignorance. The other TWIV hosts get pulled in while reviewing and help. It could be fiction like Contagion, but based on reality. I think that would be interesting. Do you think it could be interesting enough? Right? I mean, the spread of misinformation, that could make an interesting movie, and all the stuff that happens as a consequence, right? I think that would be fascinating. I'd watch it. Will says, modern smartphone tech is amazing. Some have made feature films on an iPhone, and Apple do their product release videos on their latest iPhones, and those videos are stunning, and mobile mics are great. So we have a pair of mobile mics that I used in Virginia. Oh my gosh, the sound quality is beautiful and you can barely see them and there's no wires. That's the thing. I have to carry all these wires with me. So that's why I want to do this. Visto writes, I recently heard of double-stranded DNA repair in whales that is superior to human DNA repair. That's very interesting. I wonder if they could repurpose that in some way for gene editing. Yes, what a good idea. I have to look up that. Wow, that's very cool. Oh my gosh, look who's here. Peak Dunning-Kruger. How's college, man? You can see Peak is one of our moderators, but he's wrapped up in college. It's going well here, man. We're having a good time. Nevitz writes, it's remarkable that the guide RNA can find its target out of 2 billion base pairs. Yes, that is amazing because it's all wrapped up. It's all wrapped around nucleosomes very tightly. So what does that say? Well, when the cells divide, it decondenses, I think, right? Maybe that's part of it. It's a fascinating question. Well, you could put any DNA that you wanted. Yeah. I mean, we're talking about a short piece of DNA in there, right? But yeah, anything you want. It could be from another person. Yeah. 42 days and counting, January 2026, the U.S. loses its measles elimination status. How does everyone feel about USA withdrawal from WHO? It is horrible. It is unconscionable. It is ignorant and inhumane. It should not happen. We were the biggest supporter of WHO, and we should be back there. It should happen tomorrow. It's totally crazy that we're not supporting them. It's not. This is ignorance at work. I can't say enough things about it. Peak might be in New York. If you do, come to the incubator. Vincent says it's too complicated. Oh boy. Yes. I'm amazed it works, frankly, because it's pretty baroque there what they have to do. So Vanity says 5% masks, New York and Salt Lake. Denver, New York, 10%. I don't know, LA. Not many people. I don't think it was 5%. But you know, I'm only, and none of the attendants were masked on either flight, and the pilots were not masked either on either flight. Yep. RT is notorious for its low fidelity. Yeah, it is, but it's a pretty short sequence, right? One base, it makes a mistake per 10,000 bases, so maybe you got the odds in your favor, but it's something you could study. Yeah, I agree. Is this similar to how one edits out the BRCA mutation from embryos? That would be the idea, right? You could take an ova out of a female and edit out the BRCA mutation by CRISPR and then put it back in. Yeah. Yep. An embryo, not an ova. An embryo would have to be fertilized, right? So Visto asks, would prime editing not be in danger of reverse transcribing the wrong sequence? It's possible that it could. Yeah, I mean RNAs. So this is the nucleus where this is happening, right? So it's not devoid of RNA. I mean, the RNAs are made and shipped out to the cytoplasm, but there's still RNAs in there. So yeah, it could happen. Yeah. If you corrected a muscular dystrophy gene with CRISPR, would muscle cells regenerate, or would it be too late? Yeah, it's too late. You have to give it a gene to most of the cells, right? So you have to deliver it to the whole person, and it has to be a gene making the right protein. That's currently the approach to correcting muscular dystrophy. as far as I know. That's a good question. So you can use CRISPR. So you have to give the virus carrying the CRISPR IV so it can hit as many cells as possible, as many muscle cells, and then there is regeneration of muscle from stem cells. So hopefully you can get those as well. But the more doses you get, the more better off you're going to be. Yeah. Mark says, put it in the fibroblasts, the new muscle would work. The active sarcomeres are formed. Peak says, ChatGPT has checks to prevent any sort of questions about aiding protein design whatsoever for good or bad. Oh, that's interesting. Jack Black from Mark Martin. Yeah, from Les. So apparently Bill Nye does have some acting credits, almost live. He played an FBI tech's father on a show about a girl's tattoos. Peak has many problems with lax. Getting to transport is an incredible pain. Yep. Donna wants Amy to attend the immune. She's welcome to. I don't know if she will. Do you think RNA satellite viruses exist for human RNA viruses? So let's see. So Delta is a satellite, essentially. It's complemented by hepatitis B virus. So hepatitis delta virus is essentially an RNA virus that's a satellite of Hep B. So it requires co-infection with hepatitis B virus. So yes, I mean, you can imagine defective particles becoming satellites. The defective particles can't reproduce unless they're in a cell infected with the wild type virus. So already they're kind of a satellite. They're just not called that, right? Okay, here's the whale paper from Peak. Thank you, Peak. Cristela has a casting director. He keeps saying how most of these suggestions are rest in peace. They're not going to happen. Yeah, I understand, but we're just playing anyway. How did I choose my moderators? They were people that I knew from online. So when we first started Q&A with A&V, we didn't have any moderators. And people kept coming here, and you could tell they were dependable. I saw them elsewhere. They used to listen to podcasts. Maybe they took my course, like Vanity took my course at Columbia. And so I asked them, I think some of them volunteered. Do you need a moderator, right? And I had gotten to know them. I wouldn't take a moderator out of the blue, right, who I didn't know. But I got to know them over time, and they're still here. It's great. So we have Vanity, Les, Tom, Peak, Barb UK Mac, and from San Francisco, Steph SF here with 6 mods. They're not always here at once. But they also moderate TWIV live. Some of them do anyway. So I appreciate them very much. Thank you very much. Oh, it's 10 o'clock already. Oh my gosh. Pilots were notorious mass scoffers. I guess they just kind of closed themselves up in that cockpit, right? But when they mingle, they should wear masks to protect themselves. Yeah. Has a virus ever caused an extinction? Sure, I'm sure they have, but we don't know it because this happened a long time ago in the past, right? I'm sure there are many species have been extincted by virus infections, but we didn't see them. It's only in the last 100 years that we've been studying viruses in some detail, so not happening. But I'm sure it's happened, yeah. Vincent, what did you mean in your mini lecture about a donor adding to the gene? Oh, it's a donor DNA. It didn't mean it came from someone else. It's just a piece of DNA that you put in along with the CRISPR and the guide RNA that is going to be used to repair the gap. So the Cas9 makes a double-stranded break, and it can either repair them, push the ends together, or it can use a piece of DNA to repair it. And so that DNA, I just called it donor DNA. It's not from someone else. That's what I meant by that. Yes. So also, I did my course as a live stream, and some of these individuals showed up, and that's where I found them as moderators as well. Yes. Okay, folks. It's another one. It went quick. Wow. I want to thank our moderators tonight. We had Les, we had Tom, we had Vanity, and of course, Peak Dunning-Kruger. Thank you. 10 p.m. in my time zone. I know it's different from all of you, but what am I going to do? This is where I am. It's 10 p.m. Eastern Time, U.S. We'll be back next week. Let's see. What's the date next week? I'm just going to put it on right now. It is the 26th of November. Yeah, I know it's the day before Thanksgiving, but I'm going to do it. And I bet some people show up, but I know others have family around and you have to cook and so forth. But I'll be here because I really enjoy doing this. So let me thank our mods. Did I thank our moderators? I think so. Peak Dunning-Kruger, Vanity, Les, and Tom. And thanks all of you for coming. You're always amazing. How did we do with the likes and all of that stuff? Let's see. Yeah, it's fun. We got 99 viewers and 99 likes. Thanks, everybody, for coming. Great question. Have a good week. Stay safe. Till then, be curious. Bye-bye.